Imagine you are a paleontologist, not a dino normie, but a hardened professional with decades of digging dirt and cataloging petrified rocks. You operate by a core, unassailable dogma, a fundamental law written in stone, literally. Organic material does not survive the deep time of the Mesozoic era. When we talk about a fossil, say a jawbone from an absolute unit like Tyrannosaurus rex, you are looking at rock. Every single atom of the original biological structure, the collagen, the keratin, the squishy stuff, has been replaced by minerals like silica or calcite. That process, known as diagenesis, is total. It wipes the biological slate clean. If you find proteins or blood cells in a 68 million-year-old skeleton, the accepted wisdom says you found contamination. Modern roadkill, maybe. A sloppy lab tech. Something that definitely wasn't dinosaur. For more than a century, that rule was gospel. It defined the limits of our knowledge.
We could infer dinosaur biology, their gait, their bite force, their size, but we could never study their actual biochemistry, their actual blood. Then, everything changed. In the mid-2000s, working in the famed Hell Creek Formation, that sprawling, rugged landscape of Montana and South Dakota that serves as the final resting place for the titans of the late Cretaceous, scientists made a discovery that was by every measure of classical paleontology impossible. They were examining a pristine femur bone, massive enough to choke a Honda Civic, belonging to a T-Rex specimen. This was not just a well-preserved fossil, it was the final nail in the coffin of the old paradigm. When researchers, led by Dr. Mary Schweitzer, carefully demineralized a segment of this bone, a process where strong acids dissolve the mineral structure, but theoretically leave nothing behind, they found something that shouldn't exist: tiny, pliable, branching blood vessels.
Structures that looked exactly like the microvasculature you'd find in a recently deceased ostrich or crocodile. Even more shocking, within these vessels, they found microstructures that strongly resembled red blood cells, and surrounding them, remnants of what looked like connective tissue, specifically collagen. The entire scientific community, and trust me, paleontologists are a salty, skeptical bunch, immediately screamed nightmare fuel contamination. They said the material must be biofilm or recent bacteria, or maybe just some bizarre mineral artifact masquerading as biology. This is where the story pivots from digging up rocks to high-tech molecular forensics, and how we proved that this T-Rex specimen, which died maybe 68 million years ago, still held the ghosts of its own circulatory system.
The core problem, the one that sparked years of bitter academic warfare, was proving that the detected proteins were genuinely endogenous, that is, original to the dinosaur, and not modern contamination. The stakes were ridiculously high. If these fragments were real, it meant we could finally peek into the soft tissue biology of extinct giants. If they were fake, it meant the entire research team was chasing phantoms. To settle the debate, they had to deploy technology that sounded less like geology and more like something out of a sci-fi movie. Forget carbon dating. That only goes back 50,000 years, tops. We're talking millions. We needed a definitive molecular fingerprint that screamed, "I am 68 million years old and contain iron." Enter resonance Raman spectroscopy. This isn't your grandma's microscope. Resonance Raman spectroscopy, or RRS, is a non-destructive technique that uses specialized lasers to probe molecular structures.
When the laser hits a molecule, the energy scatter changes depending on the chemical bonds and the vibrational modes within that molecule. Crucially, the researchers used a very specific tool, a green laser operating at 532 nanometers. Why 532 nanometers? Because this particular frequency is optimized to react with heme groups. Heme is the molecule in hemoglobin, the stuff that makes your blood red and transports oxygen, and it contains an atom of iron. When the 532 nanometer laser shines on genuine heme, it creates a specific, unmistakable fingerprint pattern in the scattered light, a spectral signature that is unique to the interaction between the laser and the iron center. The results were electrifying. When applied to the T-Rex fossil remnants, the RRS repeatedly registered the precise spectral signature of heme. Furthermore, they found molecular structures consistent with collagen, the primary structural protein found in animal bodies.
When they compared the spectra to modern contaminants, bacteria, or common minerals found in the burial environment, the patterns did not match. The conclusion was unavoidable and utterly mind-blowing. They had found fragments of genuine dinosaur proteins, specifically molecules associated with hemoglobin. The fundamental dogma of paleontology, that soft tissue preservation over geological timescales is impossible, was, if not fully broken, certainly bent into a question mark the size of a 5-story building. But this led to the next, arguably more fascinating question: how? How did this incredibly fragile organic material survive a stretch of time so vast it makes human civilization look like a blink of an eye? The secret, as is often the case in nature, lies in the perfect blend of destruction and protection. It's a chemical paradox.
The standard process of decomposition relies on water, bacteria, and time to break down complex molecules like proteins into their constituent amino acids, which eventually disappear entirely. But the T-Rex had a natural preservative hiding within its own blood: iron. When a creature dies, its cells burst, releasing hemoglobin. As the hemoglobin breaks down, the iron, Fe, within the heme groups is highly reactive. Researchers proposed that this iron acted as a powerful antioxidant and a molecular cross-linking agent. Here's the gross but crucial detail. The iron, released from the blood cells, immediately starts oxidizing. It rusts, essentially, turning into compounds like goethite and iron oxide hydroxide. This rust doesn't just sit there, it creates an impenetrable, mineralized barrier, a tiny, microscopic cocoon, around the remaining protein fragments. What destroys the complex biological structures, oxidation, simultaneously creates a protective, stabilizing mineral shell around what remains. This process transforms the remaining protein fragments into a stable hybrid known as a molecular fossil. The protein is no longer purely organic. It's a stable complex of organic matter encapsulated and chemically linked to mineralized iron. This composite structure is resistant to degradation by water and bacteria, allowing the tiny ghosts of T-Rex biochemistry to persist for tens of millions of years in the subterranean depths of the Cretaceous sediment. Now, before the dino normies get too excited and start talking about theme parks, this preservation mechanism is imperfect. While structural proteins like collagen are robust enough to survive in fragment form, the much more fragile molecule of DNA is not. DNA degrades rapidly, usually within a few 100,000 years, even in ideal conditions.
The science is definitive on this point.
Yes,
So,
the preservation
RIP
is
Jurassic
remarkable.
Park. No dinosaur cloning, no theme park disasters, and no probes running around your house. But what we do have is something arguably better for science: a direct window into dinosaur biology. The ability to study dinosaur metabolism directly, rather than just inferring it from bone structure, is revolutionary. We can now search for specific proteins that indicate metabolic rate, oxygen transport efficiency, and how the T-Rex might have adapted to the highly variable climate of the late Cretaceous. For decades, the hot debate was whether T-Rex was ectothermic, cold-blooded like a lizard, or endothermic, warm-blooded like a bird or mammal. Now, by analyzing the remnant hemoglobin and associated proteins, we can start to measure metabolic rates chemically. This absolute unit may have run hot, requiring massive calorie intake and possessing an energy level previously thought impossible for something so gargantuan.
Speaking of things previously thought impossible, before we jump into the startling evidence that T-Rex wasn't the lone king, let's take a quick moment for our sponsor. Today's episode is brought to you by Paleo Grub. Are you tired of modern, boring, nutrient-dead food? Do you yearn for the raw, primal energy of the late Cretaceous? Paleo Grub provides ethically sourced, 100% natural, highly calorific protein bars specially designed to mimic the energy density required by a mid-sized theropod. Whether you're chasing deadlines or escaping a triceratops stampede, Paleo Grub gives you the sustained metabolic boost you need. Warning: may cause slight feather growth and an uncontrollable urge to roar. Find Paleo Grub at paleospa.com and unleash your inner apex predator. And now, back to the giant dinosaur secrets. For decades, the standard scientific narrative placed T-Rex firmly and brutally at the very pinnacle of the late Cretaceous ecosystem. He was the undisputed heavyweight champion, the Michael Jordan of the food chain, dwarfing everything else. Any smaller tyrannosaur fossils found were simply classified as juveniles, the gangly, awkward teenagers waiting to grow into their massive roles. But again, the fossils started challenging the accepted story. In museums and field sites across North America, several smaller tyrannosaur skeletons were found. The most famous of these specimens, often called Jane, BMRP 2002.4.1, was initially viewed as a textbook case of a young T-Rex. Jane was big, but a short king compared to the adults, maybe 20 feet long. She had a leaner build and different tooth structure compared to the adults, which was simply attributed to ontogenetic changes that happen as an animal matures.
However, a cohort of skeptical paleontologists, notably those examining the famed dueling dinosaur specimen, an exquisitely preserved fossil of a small tyrannosaur locked in mortal combat with a triceratops, noticed structural inconsistencies that simply didn't line up with the juvenile T-Rex growth trajectory. Think about human children. As we grow, our proportions change, but the number of teeth remains consistent until baby teeth fall out. More importantly, the foundational structure of the skull and the location of cranial nerve openings, which dictate brain structure, do not drastically change in fundamental pattern. In these smaller specimens, researchers observed 1. Different skull proportions. The skulls were generally more slender, not just scaled-down versions of the massive, wide T-Rex skull.
Tooth count and shape. The teeth were more numerous and distinctly blade-like, optimized for slicing and rapid bleeding, rather than the massive bone-crushing pegs of the adult T-Rex, which were built like railroad spikes designed to shear through bone. 3. Neuroanatomy. Detailed scans of the endocast, the interior of the brain case, showed neural channels and cranial nerve placement that were fundamentally incompatible with the known T-Rex growth pattern. 4. Longer arms. The arms, while still comically short in the grand scheme of things, everything is a short king next to a T-Rex's body, were proportionally longer than those of T-Rex juveniles, suggesting a different hunting style. The definitive proof came through osteohistology, which is the study of bone microstructure. Just like trees, many dinosaurs laid down yearly growth rings in their bones, visible under powerful microscopes. By sectioning the tibia and fibula, scientists could accurately age the animal.
When they analyzed the small tyrannosaur specimens, they found tightly packed growth rings indicating ages of 16 to 20 years. Crucially, the external fundamental system, EFS, was present. EFS is a dense layer of bone that forms on the outer surface of the long bones when growth has slowed down or stopped entirely, signaling skeletal maturity. Finding EFS in these smaller fossils was the smoking gun. This wasn't a T-Rex teen. This was a mature adult. Conclusion: Nanotyrannus lancens was not a juvenile T-Rex. It was a separate, distinct, absolute unit in its own weight class, perhaps 20 to 25 feet long and weighing a couple of tons, massive by modern standards, but a relative short king in the late Cretaceous. This discovery flips our understanding of the final days of the dinosaurs. For millions of years, the working model was that the Hell Creek Formation was dominated by a single apex predator, T-Rex. But now, the picture becomes infinitely more complex.
Recent, highly influential research, including a pivotal 2022 study published by researchers from the University of Bath and the Natural History Museum, suggests that not only did Nanotyrannus and T-Rex coexist, but there might have been up to 3 contemporaneous large tyrannosauroid species sharing the ecological stage at the very end of the Cretaceous. The ecosystem was not a single species monarchy, it was a highly complex partitioned landscape. This arrangement, known as ecological niche partitioning, prevented direct competition for resources. Imagine the Hell Creek environment around 67 million years ago. It was a swampy, humid subtropical paradise, thick with ferns, cycads, and early flowering plants, angiosperms. Towering conifers dominated the skyline, and everywhere there was prey.
Rex, the true absolute unit, weighing in at 7 to 9 metric tons, the weight of 2 full-grown African elephants or 15 Honda Civics, was built for massive, slow, sustained power. Its teeth were perfect for crushing the thick, bony frills of Triceratops and the dense bone structures of massive duck-billed dinosaurs like Edmontosaurus. Its hunting strategy was likely focused on the largest, most heavily defended, and relatively slower prey. A powerful stalker, perhaps relying on ambush and pure, devastating physical power. The Nanotyrannus, meanwhile, was the speedster. Lighter, quicker, and with razor-sharp blade-like teeth, its niche was dedicated to medium-sized, fast-moving prey. It was the biological equivalent of a precision scalpel compared to the T-Rex's sledgehammer. Perhaps it specialized in targeting vulnerable juveniles or smaller, faster ornithomimosaurs.
This partitioning means the ecosystem was far more robust and diverse right up until the KPG impact event, countering previous theories that dinosaur diversity was already declining before the asteroid hit. Each tyrannosaur occupied a specific job slot, minimizing the chances of two absolute units fighting over the same roadkill carcass. But T-Rex was not just defined by its size or its appetite. As we peel back the mineral layers, we find that the true complexity of this creature lay in its sensory capabilities, which shattered the persistent image of the T-Rex as a pea-brained brute. For decades, the conventional wisdom depicted T-Rex as little more than a giant lumbering lizard, an animal driven purely by instinct and brute force, perhaps struggling to even see its prey. This idea, popular in early cinema, is completely wrong.
Studies based on meticulously detailed endocasts, internal molds of the brain case, which reveal the shape and proportion of the brain, show that T-Rex was, in fact, a hypersensorial hunter. Let's start with vision. The T-Rex skull was wide in the back and narrowed significantly toward the snout, resulting in eyes that faced remarkably forward. This anatomical arrangement granted the T-Rex incredibly precise binocular vision. Its depth perception was not only good, it was superior to that of modern birds of prey such as eagles or hawks. A 2017 study by paleontologist Ken Stevens demonstrated that the overlap in the T-Rex's visual field was up to 55 degrees, better than ours. This means T-Rex didn't just see a massive triceratops, it saw the precise distance to the target, allowing for devastating coordinated high-speed strikes. It was an apex predator designed not just for strength, but for calculating the perfect kill trajectory.
However, the real nightmare fuel was its sense of smell. If you look at the endocasts, the T-Rex brain allocates an astonishing amount of space to the olfactory bulbs, the structures responsible for processing scent information. These bulbs were gigantic, disproportionately large even for an animal of its size. Researchers estimate the T-Rex's sense of smell was perhaps the most powerful of any animal to ever walk the earth, rivaling or exceeding that of a modern bloodhound. It could likely detect the scent of carcasses or vulnerable prey from distances measured in kilometers. Imagine smelling a specific hamburger from 5 miles away. This powerful olfaction served two purposes. First, locating prey in the dense, humid forests of the late Cretaceous, and second, confirming the T-Rex's role as both a hunter and an opportunistic scavenger. If a rival Nanotyrannus or another predator took down an Edmontosaurus, the
Rex would smell it miles away, saunter over, and leverage its absolute unit size to intimidate the smaller predator and claim the roadkill for itself. Its nose was a biological GPS system, tuned to the scent of death and dinner. Finally, we arrive at the most controversial topic in modern T-Rex science: intelligence. For a brief, dazzling period, a 2023 study caused global headlines by claiming that T-Rex possessed a shockingly high number of neurons in its forebrain, a count comparable to that of modern baboons. This suggested a level of cognitive complexity, tool use, and potentially even sophisticated social behavior previously reserved only for intelligent mammals and birds. The idea of a 6-ton baboon was, quite frankly, terrifying. But science is self-correcting, and almost immediately, the assumptions underpinning that neuron count were scrutinized.
Subsequent research involving a massive collaboration of paleontologists, neurobiologists, and avian specialists quickly pointed out flaws in the methodology. The 2023 study had relied on scaling factors derived primarily from modern birds and primates, which have vastly different brain structures and neuron packing densities than archosaurs, crocodiles, and dinosaurs. The revised and generally accepted consensus places the T-Rex's intelligence at a highly efficient but not human-like level, Its brain organization
As you
was
can
far
see, the
more comparable
similarities
to that
between
of
the
modern
T-Rex tissue
crocodilians,
and the
which are
modern
known to
ostrich
be highly
vessels are
effective, complex
undeniable.
hunters capable of
As you can
intricate
see, the
parental care and
similarities in the
advanced
vessel
communication.
structures
The
are
T-Rex was
undeniable.
not an egghead
But
capable
the
of
danger
philosophy or
places
building
this
complex structures, but it possessed exactly the brain required to survive as a hyper-efficient apex predator.
It was spatially aware, capable of complex motor coordination for devastating attacks, and highly responsive to its environment. It was, simply put, functionally brilliant. The T-Rex we thought we knew, the slow, clumsy, pea-brained lizard, is gone. The new T-Rex, revealed through the microscope, the laser beam, and the CT scanner, is far more bizarre and sophisticated. It was a biochemical miracle, with proteins that cheated 66 million years of destruction thanks to a protective cocoon of iron rust. It was an intricately woven part of a highly complex ecological system, sharing territory with its smaller, faster, short king cousins. And it was a sensory masterpiece, combining the muscle of an absolute unit with the binocular vision of a hawk and the smell of a bloodhound. T-Rex wasn't just big. It was biologically refined, metabolically demanding, and evolutionarily perfected. It was the ultimate predator in every definition of the word. Thanks for watching, and until next time on The Lost World.